Hi, everyone, and welcome to episode 752 of People Behind the Science. I'm your host, Dr. Marie McNeely, and today we are excited to be rebroadcasting our conversation with Dr. Ken Dawson-Scully, originally released on February 11th of 2015. Listeners, Ken's research has used the fruit fly as a model system to understand how animals adapt and cope with stresses, including high temperatures, low oxygen, and high levels of free radicals, also known as oxidative stress. This work is important for improving our understanding of cellular pathways that are involved in different neurological conditions, such as oxygen deprivation in the brains of humans who have a stroke. And Ken's goal is to find new ways to maintain neural function and survival in cells that are exposed to these kinds of stresses. And in our conversation, he shared more about his life, his career, and his research. So listeners, we hope you are ready to sit back, relax, and enjoy this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to another episode of People Behind the Science. I am excited to be here today with Dr. Ken Dawson-Scully. So welcome to our show, Ken. How are you doing today?
I'm great. Thanks for having me on.
Absolutely. Well, we are delighted to chat with you. Now, listeners, Ken is an associate professor in the Department of Biological Sciences at Florida Atlantic University. He received his master's degree in neurobiology from Queen's University and his PhD in neurophysiology from the University of Toronto. Ken then conducted postdoctoral research at the University of Toronto before he joined the faculty down there at Florida Atlantic University. And Ken is here with us today to tell us all about his journey through life and science. So today, Ken, we are excited to get to know you, both as a scientist, but also as a person. So can you take these first few minutes just to tell us a little bit about what you like to do when you're not doing science?
Sure. I have a young family, a daughter who's 12 and a son who's 9. So, balancing work and family has always been very important. I've had great mentors that showed me how they do it. And so I'm always striving for that. So I would say that pretty much all of my spare time is spent being with my family and doing whatever they want to do and enjoying time with them.
Fantastic. So does your family have any particular activities that they love to do right now?
Yeah, well, my daughter's big into basketball and she's a trombonist. And my son, he's getting into the baritone and he's a big tennis player. So we're always going to practices or games or concerts. And yeah, it's great. And then obviously we live in Florida. So I would say definitely one of our most favorite pastimes as a family is hitting the beach every weekend, which is, believe it or not, a lot of Floridians don't go to the beach that often. So we've maintained that we try to go as often as we can, coming from Canada originally.
Wonderful. Yeah, you got to take advantage of that nice weather, the beautiful sand down there. So
yeah, absolutely.
Great to hear about how you and your family like to spend your time together when you're not at work. But now we're going to switch gears and dive into that work side, the scientific side. So can you tell us now, Ken, how do you describe what you do at work to someone who might not be in science?
Sure, yeah, I'd love to. So we work with the fruit fly as our model organism. And classically, I was trained as a neurophysiologist. And during my training, and when I say my training, I mean my education before my position here at Florida Atlantic University, I also went through a lot of training with behavior genetics. And I used these two techniques to help me understand how animals, how they've adapted to different types of large changes in the environment, how they can cope with it. So we know with fruit flies are insects, and they're poikilotherms, or cold-blooded animals. And so in any given day, they can be exposed to a range of temperature from 90 degrees Fahrenheit all the way down to just above freezing and still be viable animals, still interact with their environment. And we know with us as mammals, homeotherms, that, we maintain one core temperature, and just small fluctuations in that temperature lead to enormous changes in our neurophysiology and usually to our detriment. So I'm very interested in finding out what these animals, how they've adapted to these changes, for instance, with temperature, but we also look at low oxygen levels and then other types of stresses, and then see if there's a way to turn that into a potential human therapy. So, for example, high temperature, one example would be 5% of children under the age of 5, I believe, around the world will suffer from what's called a febrile seizure. And usually if I'm in a classroom and I ask people to raise their hand, there will be several hands raised of people who know someone or have experienced this phenomenon. And so these individuals, when they have a slight fever, they will go into a seizure. And so, for instance, we're looking at pathways that we use genetics to find that with a simple drug, we can change the sensitivity of the brain to these temperature changes. The same thing with low oxygen. We know that there's lots of diseases that are related to low oxygen scenarios within the brain. The most typical example is stroke. And so we use the fruit fly as a stroke model to see if there's ways. Number one, can we allow the fly to continue operating in these low oxygen environments? Number two, is there a way to prevent the cell death in these animals? We usually go from in the lab looking at behavior of the animals from my behavior genetics training all the way down to the neurophysiology, looking at synaptic transmission, both with electrophysiology, but as well optical physiology, doing bioimaging.
Very interesting. Well, this sounds like a fascinating research, really diving into these extreme neural stresses using fruit flies as a model. So fascinating research that you're doing in the lab. And, I think when I talk to scientists about their research, it's interesting to see how passionate people get about it. And I know being a person who's so excited about going to work can be challenging. And you mentioned this aspect of balancing work and family, things like that. I think there's a lot of things that scientists are trying to get done in a day. And it's often difficult to find motivation on some of these days. So do you have a particular favorite motivational quote or maybe a saying that really keeps you going?
Right. So I'll be honest with you. I never really had a motivational quote that kept me going, but I knew, being a fan of your show, that this is something you always ask in every interview. So I started milling through hundreds of quotes and trying to find the most ideal one. And the quote I actually found that I think mimics my motivational structure for wanting to do research comes from Maya Angelou, who is an African-American author, poet, dancer, actress, and unfortunately she passed away this year. But her quote is, "Success is liking yourself, liking what you do, and liking how you do it." And I think it really, in terms of a researcher, encompasses what has to motivate a researcher, that success for a researcher isn't necessarily rewarded in terms of financial return, but more loving what they do, having a passion for what they do. When they wake up in the morning, they can't wait to get to work. Sleep is merely an impediment of loving what they do. So,
right.
I think that's really encompassed it for me. But it was one of those things that you can't be told. It's something that you have to experience. And then once it grabs you, it grabs you.
Certainly. Well, I think this is a great quote to really describe the life of a scientist. So many people, like I said, are so passionate about what they're working on. They're excited to get up in the morning and go to work, but they're also excited about coming home, spending time with their family, doing the things that they love outside of science as well. So great motivational quote there. Now, I'm curious about your journey, and you mentioned some of these mentors that you've had along the way that were great for showing you this balance in life. So can you start by telling us a little bit about some of the people who've inspired you or that you really consider role models?
Sure. So, one of those journeys that I think for me, the progress through science has always been very mysterious, really pretty much through my whole training, in the sense that there was always something I didn't quite understand about the process of academia. And I was quite ignorant of it going through the process and merely going through the experiential stages. And it's one thing I try to, when I have students now in my lab, try to really promote, that they have the knowledge right up front, that I could tell them about, this is what you should expect, or this is what's going to come down the road. But, I can remember back in high school, my first love of science, actually, from almost a career point of view, was a teacher I had. His name was Michael Roth, and he was a physicist, or he was teaching me physics. I don't believe he's a physicist. I believe actually he's more of a liberal arts professor, but he had such a passion for teaching. And I believe that his whole research program now, he's at the University of Victoria as a full professor, and I haven't been in touch with him for a long time, but was how to motivate people to want to learn. And I hooked into this really quickly and really enjoyed the class he was teaching just from the way he was teaching it. Then when I went into undergraduate training, my second year university, I was exposed to another professor, and I literally had never taken a biology course. I had never taken a psychology course. I didn't even know the concept of neuroscience existed.
Oh, wow.
And I took a course with this woman, Dr. Marlene Samuelson. She's a professor at Curry College. And she was very inspiring in the way in which she taught her material about biology. So it was a two-pronged situation where, one, wow, I love the biology that I was being taught. And number two, the way in which she introduced me into that world was so fantastic that it got me hooked right off the bat. Then I ended up doing a master's degree and an undergraduate research project with a guy by the name of Professor Mel Robertson. He's at Queen's University in Canada. And he was my very first taste of independent research. I was introduced right then as an undergraduate to this concept of how animals can adapt to these huge fluctuations in temperature, how they can employ these molecular protection mechanisms to allow them to operate at these ridiculous temperatures, like at 50 degrees without any problem whatsoever. That's the part that got me hooked into independent research. I went through training with him, and it was a really fantastic experience. And it showed me everything to do about academia in terms of what life is like in the lab. I went on to University of Toronto, worked with Professor Harold Atwood, leader in the field of neuromuscular junction synaptic transmission in Canada. He had a very strong character, and when I say character, he was always striving for excellence in his lab, and it was always apparent. He had international relationships with so many different people. You know, I remember just sitting at my desk talking to Tom Sudoff and explaining my most recent project to him. And this was back in, 1999. And, you know, it's fantastic that when you're in an environment like that, that you can share your research, be proud of your research, and also have that passion for your research. And, you know, he gave me the basis of training to hopefully as well strive towards that excellence in my own research. And then I did a postdoc with Marla Sokolowski. And Marla, you know, she is an absolute leader in her field of behavior genetics. She's recognized internationally. Anywhere I go, they know who she is. And she's got a really interesting story. I know she's one of your interviewees.
Yeah, we had fun with Marla.
Yeah, she raised her children, you know, through grad school and through her early years as an assistant professor at York University. And, you know, her success continued throughout her entire career to even today. And, you know, it's one of those inspirational things that you watch happen. And it tells you that, you know, if you want to do it, you can do it. It just takes that passion and that courage to be able to strive for it. So, you know, in my scientific career, I've had these wonderful mentors and these inspirations for me to drive me into the field of academia. But, you know, my story is a little different than that. Like, that's my academic story. But when I finished my PhD, I actually had decided at that point that I didn't think I wanted to go into academia, actually.
Really?
And I actually left academia for a year to go work for a place called Olympus, Canada. And I was called a territory or management consultant, where it was a fancy name for a salesperson for scientific equipment to universities and hospitals. And we sold high-end laser confocal microscopes and everything else. And it was something where, because I never really had a job in industry, I was in academics my whole life up until that point, that I was thinking, you know, it's time I got to give this a try. And, you know, when I graduated with my PhD, I actually had my daughter then. She was about 3 years old, maybe 2 years old. And, you know, I was also thinking about, okay, I've got to provide for my family. And sometimes when you go down the road of academics, it doesn't pay that high,
right,
as you can imagine. So it was one of those things that I definitely wanted to give a shot. And I got to tell you, the first year I worked for Olympus, Canada, back then it used to be called Carson, the people I worked with were absolutely amazing. The job was wonderful. It was very engaging, and, you know, it was very manageable with the family. I was making a six-figure income right out of the gates, out of a PhD. But within 8 months of that position, I could feel already something dying within inside of me. It was the passion I always keep mentioning, and that was the ability to create and go your own way and explore your own questions. You know, I think when you're locked into kind of a service industry or a type of industry where you have very direct tasks that you have to accomplish, you need another outlet. And I didn't have, all my hobbies at the time were science, you know, before this experience. So I didn't have any hobbies outside of my work to satisfy those needs. And I had actually called Marla, I think, 8 months into this position at Olympus, Canada. And we had known each other from before. I had already potentially arranged to go into her lab out of my PhD. And she graciously accepted me into her lab. And, you know, from that point on, making that decision, that's when I decided success for me was doing what I loved rather than the amount of money I would make. And I was committed at that point to do my best to succeed in academia and get my own lab.
Well, I think you brought up so many great points, Ken, when talking about this amazing journey you've had through science. And I think you're absolutely right when you said that a lot of people don't really understand the process of becoming an academic scientist. And that's something where I myself was totally in the same boat. You know, I knew I wanted to be a scientist of some sort, but beyond going to college, I really didn't know what happened next. So great point that you brought up there. And I think this is a great opportunity to show scientists, you know, or future scientists what the process is. So great that you brought that point up. And then also, I really like your description of going into the industry world. It's something where you just wanted to try it, but then you found out that maybe it wasn't exactly for you. It wasn't where your passions were. It wasn't what you were most excited about. And I think I had a similar experience where after my PhD, I went into the industry world because I figured may as well try it, you know, see what's out there, see what other jobs scientists could potentially get and do. But eventually wound up back in academia as well. So certainly.
It always sucks you back in.
It does. You start to feel like you're missing part of what you had when you were in graduate school. So wonderful to hear your journey through science. And I think we've talked about a lot of the wonderful aspects of journeys through science and your career in particular, some of the great projects that you're working on. But we haven't yet chatted about some of the struggles. And I think every scientist has these points in their career where they're really struggling with something. They're facing some major obstacles, and it might go on for days, weeks, even longer sometimes in some cases. And a lot of people don't hear about these situations. So can you take us back to a point in your career where you were struggling mightily with something and tell us what happened and how you got through it?
Sure, yeah. You know, I think in science, in research, these type of struggles exist on so many different levels in so many different ways. You know, I think the life of a researcher, being it a trainee or even a primary investigator, principal investigator, you're always confronted with so many different challenges. You know, the typical challenge is time management, trying to get through a set of experiments that you had planned for and wanted to do, and then balancing everything around that, or having things not work and you know, doing it over and over again and it's still not working. And you already know that this is like the typical definition of insanity, where you're doing the same thing over and over again, expecting different results. But, you know, it's kind of ground into you that there's human error involved and there's something you're not doing right. And meanwhile, there has to be a time where you accept, okay, I need to change something. But with research, you can't change things after an N of one. You've got to keep pushing and making sure that you're sure that things aren't going to work. That being said, for my own personal, I guess, moment where I could tell you that I had a struggle with was, I would say during my PhD, my first year in the lab, I was trying to figure out, you know, what was the question that was going to drive that passion and that motivation. And, you know, in the beginning, the project that I was on wasn't really doing it for me. And I got very lucky in the sense that, you know, I was rescued out of that scenario by a collaborator of ours. Conrad Zinsmeyer is a professor in Arizona, and he wanted to look at something with a technique I was using at the time, and I really wanted to become proficient at. And I was lucky in that sense. But this also leads into another issue, and that was when I did the initial experiments, and these are a number of manuscripts from my PhD where I was working with a protein called cysteine string protein. We made a finding from our experiments that was absolutely opposite of what was published.
Really?
And this was really troubling because, you know, the struggle for me was that everyone was looking at me that I did something wrong or I'm not doing it correctly. And the struggle for them is that after I did the experiments 100 times and the exact same experiment was yielding the same results from a number of different angles, that we had to bite the bullet and actually publish that work. And, you know, there were other great scientists who I admire who had published the initial results.
Oh, wow. So how did that go?
Yeah, this led to a lot of uncomfortable moments, especially at conferences and even seminars that I gave at my university or other places where people were on the other side of the coin saying, you know, this is probably wrong. What
so
about
you were not
results?
The popular guy in grad school, is this what you're saying?
Yeah, well, no, with some people. But, you know, it was really fantastic experience because I got to learn what the academic process was all about by going through that experience. And that is, you're going to publish your results. And the point of publishing them is so that they're challenged and that, you know, it's funny how we relate these days a hypothesis versus a theory, right? We're still at the level of a hypothesis, even when things are published, even when the data looks very strong that this is what's happening, because it's out there in the public eye, not only to be disseminated to the public, but also to be challenged. And, that's in essence what we did. We went to repeat the experiment and we found different results, and then we ended up going to this position of challenging. And, at the end of the day, I wouldn't say that a lot of it was worked out. From my perspective, there's probably some technical errors that were done on the other end of things, and there were other publications to support that. But, I think our work is still referenced in the work done on cysteine string protein moving forward. But it also, led to some, like you said, very uncomfortable scenarios. The one great thing about that experience, because it did lead to obviously a lot of little negative things as well, but the one great thing was that my PI at the time, Harold Atwood, defended my work in public, was always behind me, had my back. And that was really important for me in such a junior position, because without that, it could have been crippling to my confidence and courage moving forward.
Certainly. Well, I think having that mentor supporting you is really important, not only in these times of extreme struggle, but just really throughout that graduate process. So wonderful to hear that you had a great mentor who had your back during this tough time. And I think you really brought up a great point there in terms of science being dynamic. I mean, this process of critiquing and challenging what's out there is so integral to the process of science. And it's something that I think you forget when you're maybe the person who's challenging what's out there. But, science is always changing. When you look at things that we believed 50 years ago, how many of them are still true, sort of deal. So,
right. And I find it shocking, like even in public media. And, I talk about this with my colleagues here now all the time. And that is, the difference between a hypothesis and a theory, that the public media engine always uses theory as some sort of negative word. Oh, it's just a theory. But by the time things reach the level of a theory, there are unsurmountable amount of data to support that theory by the time it actually becomes that. So a theory is a very strong position compared to a hypothesis.
Exactly.
It's shocking, yeah.
Well, great to hear about how you worked through this tough time and that there was sort of a silver lining to that dark cloud in your career. So great to hear about one of these challenging times, but we don't just want to focus on the tough times in your career, Ken. Can you tell us next about a success story, one of these moments that really meant a lot to you? And it can be a big one or even just one of these little victories along the way.
Sure, yeah. So I would say that would probably be during my postdoctoral fellowship and the beginning of my own research program that continues on today. Having a mentor like Marla Sokolowski was extraordinarily supportive for me developing my own research program. There were when I went into her lab to look at specific questions to do with the foraging gene and to understand the physiology or the physiological basis behind the behaviors that she observes with the different feeding patterns in these two alleles of this gene. So the two versions of this gene. One feeds a lot and moves a lot, and then one doesn't move very much. But she let me actually develop a side project where I developed my own theories about how this gene might actually control neuroprotection. And when it comes to neuroprotection, I started very early working with a molecular mechanism that was genetically controlled called the heat shock effect. And this was mediated by heat shock proteins. So there are these proteins that are in our genome, that are in genomes of the prokaryote, and pretty much all life on Earth. There are a few species that are missing it, but when they undergo a stress, and that could be with temperature, that can be with oxidative stress, some sort of toxin, low oxygen, for instance, even exercise in humans, that we actually turn on the transcription and translation, the production of this heat shock protein. And it selectively is produced when stress is present. And the interesting thing about this protein is that when it's put into, just say, a simple vial filled with a protein that's denatured, say something that would normally glow like luciferase, it will fold it back into its native state. And this was one of the concepts that I carried with me from my undergraduate training all the way to what I do now. But when I was with Marla Sokolowski, a manuscript was published, and this was by Chengfang Wu's, where they showed that there was an actual physiological change due to the foraging gene. That is, when there was less of the foraging gene present or less activity of the product of that gene, this protein PKG, protein kinase G, that the potassium currents in the cells would be a very specific pattern, and they were very reduced. Where in high amounts of activity and high amounts of this gene product, the potassium channels would be very open. And in that exact same year, another paper was published out of the lab that I did my master's degree in my undergraduate research with Mel Robertson, where they showed the exact same pattern with animals that had been heat shocked and non-heat shocked. So I was able to actually go after a question looking at the differences in the foraging gene for neuroprotection with the hypothesis laid out that low PKG expression, so this protein, would lead to neuroprotection from high temperature stress. And it was kind of like the true academic process that you go through the previous literature, you come up with a plan and a hypothesis that you can test, and then you test it, and it actually worked out. I remember feeling so excited when that did work out. It was very moving at the time, and rightfully so. It's what I pursue now as a PI. Like, it was one of those things that without that opportunity to go through that, I might not have the independent research program that I do now.
Definitely. Well, I love this almost textbook case of science at its finest, working well. Things happen as you expected them. And I think oftentimes that's a rare thing to come by. So great to hear that you've had this experience.
But you did ask me to find one instance of it, and there it is.
Yeah. So has it happened since? Something so beautifully laid out, put together, and then the results look great.
Yeah, it's a hit and miss. Like, I find that with science, you always have a plan to move forward, and a lot of the time you're surprised. And I think that's part of the addiction to science, and that is that as much as you plan for something, it might work or it might not work. But I would say that most of the time in those scenarios, that's not clearly the answer, that maybe it doesn't work the way in which you planned, but all of a sudden all these other things came out of that experiment that lead you down a different road. And you're always chasing these ideas that experiment led you to. And it's fantastic. I think it's part of why I love science to begin with.
Certainly. I think that unpredictability, that being surprised, that getting something that makes you stop and go, wait, what happened and why? I think that is part of why people get so addicted to science. I think you're absolutely right there. So I think researchers definitely are so invested in their research. They spend quite a bit of time doing it, but they also like to do things for fun. Like you mentioned, spending time with your family. A lot of researchers are spending quite a bit of their time reading as well. Not only the literature, the things for science, but also some fun reading as well. So I love getting book recommendations from people we have on our show. So can you share with us today a book that you've read and really enjoyed that you think we should check out?
Sure. So I usually read fiction. I read enough nonfiction with all the literature, and I usually stick to either fantasy or dystopian books. And I think it's for me to try to get my imagination going. I think imagination is a really important facet of getting new ideas, developing innovation. Staying creative. Yep. Yeah, and so one of my favorite authors is this guy, Brandon Sanderson. And, if I was to recommend a book to you, I would say, the Mistborn trilogy. But anything that guy has written is just fantastic. And it covers pretty much the breadth of what I'm interested in, from fantasy all the way up into dystopian futures. But a book I've actually been reading recently, which I'm surprised that I'm really enjoying it, and it's a really powerful book, is this Atlas Shrugged from Ayn Rand. I know this book's been around forever, and I've just never picked it up and read it before. And I find it fascinating, and I find a lot of interesting things that I can now understand why there's certain popular media references to it, which I think is hilarious, because I think one of the key terms in the book is always check your premises. People always say that in the book. And so I think it's a book that you could pull pretty much anything out of. Little one-liners here and there, yeah. Yeah, depending on what your premise is, which is very funny. But the part I'm really enjoying about the book is the fact that it's one of the few fiction stories where I see how science or innovation is so highly regarded as the engine for all innovation. And the times that we're in right now, where federal funding is so cut to fund the sciences, I find it like a very interesting read that all the potential negatives that come from cutting science will lead to. Those are excellent recommendations, Ken. I'm going to put those on our website for our listeners to check out if they're looking for something fun to read to escape maybe from the literature for just a little while. Yeah. So great recommendations there. And I know we've talked about a lot of great things about careers in science. We've mentioned just the way that you can work on things that you're excited about, that you're passionate about, that you're looking forward to get to in the morning. But I think one of the other wonderful aspects of careers in science are some of the opportunities that you get to travel, to share your research. So can you think back now on all the places that you've been for science, Ken? And can you tell us about your favorite place? And what was it about this experience that really just made it so memorable? Sure. So I'm at Florida Atlantic University right now. And I'll be honest with you, before I even came to Florida Atlantic University, I've never even heard of it before. So I was in Toronto. I was always looking for Canadian jobs. And there was a job ad, I believe it was in Science or Nature, I can't remember which, was a job ad that matched my skill set perfectly. And the chair of the Department of Biological Sciences is a guy named Rod Murphy, and he still is. And I knew Rod from all the different conferences that I've been to, all the literature I read about insect physiology. Like, he was a big leader in the world. So I actually came here and applied here because he was here. And coming here was an interesting experience because when I came to interview, I didn't realize how large this campus was. 30,000 people. There were like so many different departments, at least 4 neuroscience departments that were dedicated to neuroscientific work. And then all of my biological sciences colleagues, so many of them working on the biology of stress. And that was a really fantastic experience. But one of the things that I was also very surprised at was that my campus is actually the site for the first North American Max Planck Institute, which is literally, I could throw a baseball and hit the building right from the building I'm in right now.
Oh, wow. I didn't realize that either.
Yeah. And on the other side of my building is the second Scripps Research Institute. So these 2 institutes are on my campus. The Max Planck Institute is actually dedicated completely to neuroscience, and Scripps has 5 departments in it, and one of them is a neuroscience department as well. So in the environment I'm in, I could never imagine being a part of this. And, Scripps and Max Planck and Florida Atlantic University have this fantastic relationship where it's like one campus. I have access to all of their facilities. They have access to all of ours. So it's one of those unique opportunities. And so to get back to your question, and I had to put it into perspective, I'm sorry, is that because Max Planck was here, Max Planck doesn't have its own educational program. It's a research institute. And so the way it works in Europe is that a Max Planck Research Institute will couple with a university, and that will become their educational counterpart. And when Max Planck first came here a few years ago, I think it was about 4 years ago, we actually had this opportunity to meet all the people that were being hired for Max Planck and then go on a joint trip with them to an example institute where they have 5 Max Planck institutes at the University of Göttingen in Göttingen, Germany. So Göttingen, Germany is right dead center in Germany. It's 2 hours north from Frankfurt, 2 hours south from Hanover, and it's one of the oldest research institutes in Germany. And it's even got a cemetery with, I think, over maybe 1 or 2 dozen Nobel laureates. It's had the most Nobel laureates at that institute. And when we went for a tour of how the university and Max Planck interact in Göttingen, we got to meet all of these fantastic neuroscientists, Reinhard Jung, Stefan Hell, who just won the Nobel Prize this year, Erwin Nair, who won the Nobel Prize in 1991, so many other good people, so many great students. It was definitely one of the more memorable trips that I've ever had in my life. And the great part about that is we also met a couple of representatives that run the educational piece there that we're working with right now to build a joint exchange program between our triad up here, between Scripps, Max Planck, and FAU, with the University of Göttingen and the Max Planck Institute. So we actually just came back from our second trip from there and piggybacking a conference at that institute. And it's one of those things where I got to go back a second time, and I can't wait to go back a third and a fourth and a fifth. I think we're building up a really great relationship, but it's definitely one of the more memorable scientific trips I've made.
Well, that sounds like an incredible travel opportunity, and phenomenal that you have all these amazing resources and colleagues right there, just sort of right next door to your university. That's amazing. So great to hear about your opportunity to go to Germany. So what was it like, when you first went there? Was this your first time to Germany?
So the first time was a few years ago, and that was my first time to Germany, first time with the culture, and it was fantastic. It was very different. I didn't realize every German breakfast, there's a candle on the table, there's a soft-boiled egg, there's a basket of bread and a plate of meats and cheeses, and the culture is very different but very similar, and it was a really interesting trip. I really enjoyed it. I think one of the things I definitely want to do is be able to bring my whole family there and enjoy it with them as well.
Certainly. So was there a favorite site or a favorite place that you visited in one of the institutes or around the area when you were in Germany?
Just being able to tour the labs of the people I mentioned, like walking into Reinhard's department and seeing his laboratory space and his office and
seeing where the magic happens. Yeah.
Yeah. And just seeing the actual facilities they had, it makes you look at, what things would be like if you had the right budget and the right people. And it's such a fantastic experience. You could see that the Germans invest so much into their science. Obviously, though, this is like one of the top institutes probably in Germany. So, it's kind of, it's a bit unbalanced from my perspective, but it was really fantastic to see it and see these people. I think the laboratories were my greatest sightseeing aspect of that trip. But that being said, obviously, there were lots of beautiful things and everything else.
Spoken like a true scientist there. I like it. The labs were phenomenal.
Exactly.
Well, Ken, great to hear about this opportunity and exciting to hear about how this opportunity is continuing in terms of building that collaboration with those institutes over in Germany. So best of luck on continuing that collaboration and some future visits over there as well. Hopefully you will be able to bring your family along.
Yeah, that'd be great.
Definitely. Well, I know I love chatting with scientists about these travel opportunities. And I think that's something that a lot of people, when they're thinking about careers and maybe thinking about careers in science, don't realize these opportunities are available. So we love to highlight them on our program. And in addition to some of the great places you get to go, I think you get to meet a lot of great people. We've talked about some of these people you were able to meet in Germany, as well as people that you've worked with over the years, some of the incredible mentors that you've had. And I think when you work with people so closely for so long in lab groups and with your mentors, you get some quirky traditions that pop up or just some funny memories that you can look back on. I think scientists are very creative people. So you get some interesting things that come along through the years together. So can you share with us today one of these quirky traditions or a funny memory of yours?
Sure. So in my lab, we don't really have any quirky conditions.
You'll have to work on that.
Yeah, I think maybe as I get older, I'll fall into something like that. But when I was doing my PhD in Harold Atwood's lab, every Wednesday, I believe it was, at 3 p.m., there would be something called tea time. And they had a list where everyone had to alternate and bring in a cake. And so you'd bring in a cake and everyone would meet around this lab bench and we would all get a piece of cake and, bring in our coffee, or in my case back then it was a Diet Coke, and generally I guess it was tea back in the day. And so it was called tea time. And I thought that was great. As long as Harold Atwood had that lab, and he's retired now, at least from having his own lab. I know he's quite active still with working with grants and working within the university, but he had tea time every Wednesday at 3 o'clock. I thought that was pretty fascinating. It was quirky because we would always try to throw a wrench into it somehow. Like I remember one cake time I brought in an enormous gingerbread house that I think only got half eaten, and then the rest of it got put on top of a refrigerator for the next, I think it was about 8 years before it actually disappeared.
Oh, wow.
It made it right to the end of the lab's lifetime. So it was funny moments like that allow you to reflect on, not only the comedy of it, but then also the great social interactions that you have, and you chat with other people and get to know them and build relationships.
So what did you guys chat about in tea time? Was this for talking science or was this more for just hanging out?
It was more for hanging out. I remember getting questioned about, because we were in Canada at the time, on Canadian history and being chided for my low education in Canadian history, but we would all joke around and laugh. It was far more social. We had obviously lab meetings every week and the less social aspect of the interactions, but I think that Harold Atwood always wanted that piece to be a social piece, to be a relaxed piece, and people can mill around and joke around, but it was important that he always brought them together. And I thought that was pretty admirable. I thought that was a great thing to do.
Well, I think that sounds like a marvelous tradition to have in the lab, to bring people together, have some food. I love this idea of the giant gingerbread house. It sounds incredible.
Yeah.
So, Ken, do you see tea time in your lab's future? Is this something that you might spring up in the later years?
I don't know. My lab meetings are a little different than the lab meetings I've been exposed to in my past. I think that kind of interaction where you bring everyone together, I think it's different for every lab. These labs are like families. So for my lab or my family, it's got a bit of a different spin on it that, we'll have like our business as usual. What do we need to do? We'll either go through a manuscript or data, or someone practicing for a seminar, or want to go over their poster. And then there'll be a kind of a social aspect afterwards. But these things I'm sure evolve over time, and this is where it is right now. It's been a lot of change with my lab. My lab used to be in Boca Raton for the first 4.5 years that I was at FAU, but then we moved our whole lab up to Jupiter, and that's where Scripps and Max Planck are, so we're right beside it. So there's been a lot of disruption as well. So I don't know if we've had the years to settle into a good tradition yet.
Gotcha. Well, there's still plenty of time, Ken. I love that tradition. So great to hear about some of the funny memories, some of the bonding moments in your lab that you've participated in previously. And I love talking to scientists about these opportunities to have fun in science. And I think it's great to have that culture building in your lab. I think when you're talking about different lab meetings or different opportunities where the groups are getting together, you talk about some of these big questions, some of these things that you would love to answer if you had all the resources, everything you could possibly ask for in science. So can you tell us today, Ken, if we gave you everything, if things like funding and staff and technology and feasibility were not an issue, what is one question that you are just dying to know the answer to?
Well, it's a tough question, to be honest. I don't know if you have like these large pauses right after you ask such a question, but
they may come in now and then.
Yeah. So, I think over the past year, there's been one aspect of my research that's kind of gone a different way. And that is we've been looking at epilepsy models in our animal, in the fruit fly, and using electroconvulsive seizure as inducing a seizure in the animal and looking at recovery time. And this was something that was done, I believe it was developed by Richard Baines, who's at the University of Manchester. And it actually mimics what happens to the adults when you do the electroconvulsive seizure in the larval stage. And for a while, we were playing around with this genetically, and it was working beautifully, but we were trying to also work with drugs. One of the things with epilepsy is that every year there's fewer and fewer drugs that are available. Another statistic with epilepsy is that the efficacy of one drug for epilepsy, using one drug, is about 60% it'll work. But the exact same efficacy was available in 1897 with potassium bromide. So it's kind of disturbing in that sense, and that a lot of the therapies that are used for seizure now for epilepsy are these multi-therapies. And sadly, we've lost young friends personally from an epilepsy disorder. So it's one of those things when we went after looking at this epilepsy model in fruit fly and using different levels of the foraging gene, we actually found this beautiful difference, but we couldn't mimic it using drugs. And this is obviously a really key feature because I think if you build a model, you want to be able to develop it to do drug screens. Like, there's all these high throughput systems available nowadays through Scripps Research Center. The NIH has funded the Sanford Burnham, and they can screen 1 million small molecules with a robot using 96 or 1,100, whatever, well plates for cell-based systems. And, one of my dreams would be able to utilize one of these for an epilepsy model, obviously in an in vivo animal-based system. And actually, we collaborated with Kailiang Jia, who's one of my colleagues in biological sciences, to develop this fantastic model where now we use the C. elegans nematode worm, and we use the exact same genetic backgrounds, approaching the same question that we did in the fly, and got the same results.
Wow.
A, that was great. That was fantastic. But B, with C. elegans, we can actually add drugs now into the solution where the C. elegans just moving around for 30 minutes, and we get the effect of the drug as well. So we can recapitulate what we found genetically using drugs. And so back to your question, you asked me what I do. I would want to develop a high throughput screening system by putting one C. elegans in every well of one of these plates that can be handled by a robot and be able to screen the millions of small molecules that are out there to be able to find new therapies for this and be able to get to the neurophysiological basis of how they work using our lab. So that's what I would want to do.
Well, that is an excellent question. I like that you've already got the approach worked out. You've got all the details put together. So what's holding you back now? Is it funding, staff, technology, or feasibility? Which one of these do you think is holding you back the most?
Well, nothing's holding us back. We're moving forward no matter what, but we're moving forward at a glacier pace.
Right.
It would be nice to be able to speed up that glacier somehow. And I think, unfortunately, the way in which science works is money would be the thing that would speed that up. But we're actually funded by a private company. One of the things that we didn't really go into, and I really didn't offer until this point, is the fact that once I started my postdoc work with Marla Sokolowski, we started commercializing our findings. And a lot of the stuff we found with fruit flies, we found obviously potential medical uses for humans, and we started patenting those works. And we have a patent portfolio now that is licensed to a private company that actually funds the lab. So we actually have funding for the lab to do this work through the funding that was obtained by the licensed patent portfolio.
Very interesting. So how did you realize that you had something that could be patented? I think this is something a lot of scientists maybe consider or maybe is completely off their radar, this idea of patenting some of the technology that they're working on.
Right. Well, there's always two arguments for and against patenting when it comes to science. I don't think I would want to take sides on which side is right, but one thing that's evident in science today is the lack of funding.
Mm-hmm.
And one potential avenue to gain funding is to actually turn your findings into property that you can license out. And so for us, the way I started going down this road was I was in Marla's lab, and we made this finding with heat stress and the tie-in with febrile seizure in children. And then there was also obviously all these other potential applications for firemen or military to prevent febrile seizure from taking hold due to strenuous environment. And it's even been, suggested maybe with athletes. But one of the things that when I was in Marla's lab was someone from the technology transfer office was trying to see if there was anything available. And, when you sell your science story, especially nowadays, it's very hard to sell pure science. They always want to see some sort of medical outcome from what you're doing.
An application, yeah.
And so if there's a tool involved, then it's not necessary. But if there's actually some sort of scientific question where you're looking at a particular molecular pathway, there's got to be this medical spin. And since I've been training, it's always been that way because the funding structure is such that the higher funding is from medical sources. And so we had this spin on the story. And when we met the tech transfer individual, they said, well, we should be patenting this before you publish it. And so we went through that process with them. And the first patent was obtained at the University of Toronto. And then when I came to Florida Atlantic University, we made another finding, and then we got a second patent, and then another one for stroke, and then we have a patent pending for a third patent. So, it was building up a portfolio, but this means nothing unless you can do something with it.
Right.
And so from our perspective, what we did was we built a short marketing package. I have a colleague here at FAU. His name's Ram Narayanan, and he's a professor in biological sciences, and he's had experience in this. And so he helped me put together a small marketing package that he helped me distribute to all the big pharma and the small pharma companies that were local. And from the responses we got from big pharma, this is too early, we're not interested. But we got a couple hits from small pharma. I met one group, and, this is ironic, they're from Canada.
Oh, wow.
And the CEO of a company called Pivotal Therapeutics, who had an investor that was interested in spinning off another company that ended up being our company that licensed our work called Neurobiology. So we ended up licensing the patents to them, and we've been funded for about a year and a half through that revenue stream. It's been great.
Well, that sounds like a wonderful experience. And I think interesting to get this angle on funding. I think you're absolutely right. Right now, funding is difficult to obtain. And it's one of the things that a lot of scientists are listing as a pain point, something that they are struggling with right now or have struggled with in the recent past. So interesting to hear another funding option out there to go this route and start patenting some of your work and see what sort of collaborations you can form there. So very interesting. And now, Ken, I think we've had some exciting stories shared from you today, and we've had some great conversations. And we're coming to a close now. So is there any last piece of advice or maybe a last piece of inspiration that you want to leave our listeners with today?
Sure. So I can tell you that when I first got into science, obviously I had a predisposition to be really passionate about the work I was doing, and I really enjoyed it. And I really committed to that, I think, when I entered into my postdoctoral phase. And, it was always my idea that anyone can do science, that as long as you have the drive, the tenacity, the interest, and the tolerance for the mundane, which means that, you're doing experiments where, for example, you're watching fruit flies pass out from low oxygen or high temperature over and over and over again.
I love that tolerance for the mundane.
Right. So there's got to be a bigger question that makes you interested in doing that. And it's something that I try to tell all of my students or anyone who wants to come and try research or work in my lab. And that is, unless that passion for that question is there as a layer, all the rest of it's not going to fit into place. And then it led me to also understand, unfortunately, that not everyone is built for science, that if they can't build that situation where they can be passionate about the question and do the mundane to get at that question, then it's not the place for them. And I know that sounds negative, but it also, I think, opens up the possibility that maybe my science isn't what you're interested in, that to try as many different things as you can to find that question. And that is, if you're interested in research, but you're just missing the question, then go and figure out all the different questions that you could potentially try to answer. You can only do that by getting the experiences from as many different laboratories as possible. So that would be my inspirational suggestion, is that if you're interested in research, to figure out what you're going to fall in love with before you really try to commit yourself, because otherwise I think it sets you up for the mundane and it'll ruin your perspective on what research is all about.
I think that was very well said. And I love this idea, this feeling, this belief that anyone can do science. You just have to have that almost a long-term vision and excitement about the question. So great message to leave our listeners with today. Now, Ken, can you tell them how they can reach out to learn a little bit more about you or your research?
Sure. I think if you just Google my name, Dawson, Dawson -Scully, Stull, all of the faculty pages and press of the company, and I think there's even some seminars I've given in the past up there. I think the top link will be a lab webpage that they can feel free to utilize any of that contact information to get a hold of me.
Wonderful. Well, listeners, feel free to check out that website and see if you have any questions for Ken. Definitely reach out, get in touch with him. And Ken, thank you so much for joining us today and sharing your story.
Oh, it's a pleasure. And thank you for having me. It's been great chatting with you.
Well, thank you so much. I've enjoyed our time together as well. And listeners out there, thank you too. And join us again next time for another episode of People Behind the Science. Your voyage to the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guests' favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to seeing you next time on People Behind the Science.